All right, good morning, everybody. Hello, hello, hello. As the best London policeman would say, what's going on here? All right, so we left off partway through our numbered step plan for chi-squared success. So we're going to continue on that, and then we'll do a couple of practices before moving on to mitosis. Sorry, shouldn't get those two confused. Okay, so I suppose I better stand up. Get off my butt. Sit down too much as it is. Okay, so no, they're not rules. Process, I guess. All right, before I forget, let me bring up the chi-squared test table. Ba-dum. Pardon me. And you'll find this in the exams and quizzes folder, which is what I'm just opening up right now. All right, cool. So we started on this on Thursday, we just didn't finish it. So first step is what? Haha, quiz for the class. What's the first step in a chi-squared test? Most important one of all. Without which the rest of it is largely meaningless. You think you just get to sit there and listen to me chunter on? Honestly, no. Think. What's the first step? What do you need before you can do anything else in the chi-squared test? Begins with H and ends with is. Yeah, hypothesis. Right? Why do you need the hypothesis? What do you get from having a hypothesis? Good job, Amanda. Exactly.
It gives you your probabilities.
Exactly. Good stuff, whoever that was. I don't, it's like the who is speaking pops up so quickly, I don't always catch it. Yes, it gives you your probabilities. And from your probabilities, then you can calculate your expected values, right? And so that's why it's so important. Hang on a second. Oh, it's a scam likely. I love my phone. Isn't that so cool? Like before, I just ignore every phone call that I didn't recognize. Now it actually tells me it's a scam. It's like, yeah, it's from Pennsylvania. Always. I don't know if that's like the new Nigeria or something like that, but anyway. So figure out your, actually, let's do it formally. Design your hypothesis. I mean, technically speaking, you really should have figured that out before you start the experiment, but we'll just forget that kind of detail right now. So design your hypothesis, because that gives you, as someone said, probabilities, e.g. F2 monohybrid equals, and I'll do it as a fractions rather than a ratio because it makes a more sense in this case. Right, so that gives us our, sorry, a bit too much of an angle there, our probabilities. What's next? In our multi-step plan for chi-squared success. What do we do with those probabilities? What does that allow us to calculate? Yeah, expected values. Thank you, Amanda. Right. And so, again, as I'm pretty sure I mentioned this on Thursday, one of the classic mistakes students, sorry, plural, make, is that the expected values are calculated using the total observed values, right? E.g. the dominant phenotype in a F2 monohybrid equals total observed times probability. E.g. 3/4. Hello, Jess. Okay. So for example, actually, let's pull one from the PowerPoint. Okay, so, crap, that's a big number. From which one are we on? Slide 9 in the probability and statistics PowerPoint. We're going to use the top one, right? Which is round versus wrinkled. So we have, what's that slide? 9. Top example. The total is 5474. Can you all see? Actually, let me just pull this up on shared screen. So you know what I'm looking at. I'll get off that in a second. All right, so it's this top one here, right? In that PowerPoint. That's what we're going to use. So the total observed is the total of both phenotypes observed. If we're dealing with a monohybrid cross and 2 different phenotypes, obviously if it's dihybrid cross, there'll be 4. So the total is that, which is 4, 6, 7. That's 7, 3, 2, 4. Showing you my mad mathematical skills. That's about the limit of it, to be honest. Right. So if we want to figure out how many dominant individuals we would expect, right? Not that we're going to get, what we got is this number here, right? That's the observed. What we think we should get is the total, not the dominant, but the dominant and the recessive added together. So the total times the probability of getting that expected phenotype. Critical to get this right. Otherwise, your chi-squared test will be wrong. Guaranteed. Pardon me. Hoopy-doopy-doo. Got to get the old calculator up because I can't do that just off the top of my head. What's that? 7324 times 0.75. It's 5493. Okay, so, oh, hey, thanks, Lindsay. What's the expected value, or how many recessive individuals would you expect? Right, I'm going to leave you to do that. How many recessive individuals would you expect in a monohybrid cross where we observe these numbers? How many recessive individuals? All right, Lindsay, could you show your workings? I'm assuming that's right. itchy nose. Yeah, awesome, thank you. So it's the same total, right? Because you work with the same total. Doesn't matter how many different individual classes or phenotypes we've got. You use the same total for every single one of them, right? And so the other probability, the probability of getting a recessive, I should probably add that just to make things crystal clear. Is the total times quarter, which, as Lindsay put, is 1831. Okay, so these are our expected. Oops. My spelling function is having to make space for my mathematical function this morning. Those are our expected values. All right, everybody up to speed? Does that all make sense? Again, just to reiterate to the point of being annoying, always work with the total number of observations. Because you will have to do at least one of these in the exam. And it takes time. Not a lot of time if you're good at it and you practice, but it does take time. Okay, that's step 2. Step 3 is calculate the chi-square value. Right, that's the next step. I'm gonna have to put this a little bit lower. If I'm pointing like some random direction, just let me know and I'll correct it. So the chi-square value is calculated as such. It is the sum of come across that in lab already, right? That funky E. The observed minus the expected squared over the expected. So essentially what this means, to kind of pass it out, is for every single class of whatever you're observing, you work out the difference between what you expect and what you observe. And the reason why you divide it by the expected is you need to take into account the size of those values, right? Because obviously the difference between observed and expected, observed here, expected here, observed here. Let's just get that out of the way. I'll confuse things. That actual number is going to be larger than if your total observations were, say, 100, right? Like in absolute terms, the difference between the observed and the expected would be larger. But what you care about is not the absolute difference, but like the relative difference. Does that make sense? So you have to take into account the numbers that you're working with by dividing it by the expected value. And the reason why you square it is the same as for variance. You're going to have potentially, well, you will have numbers which are higher than the expected and which are lower than expected, right? So this basically turns them all into positives. And so you do this calculation for every single variable or condition or whatever that you're observing. So we're doing a dihybrid cross. We have 2 different phenotypes, conditions. So we do 2 of these calculations. That's what the sum means. You add those together. right? And so as with what we showed in Excel, you do the function within the brackets or the parentheses first, and then you square it. So this is going to be observed for the dominant minus the expected squared expected dominant plus observed recessive minus expected recessive squared, pardon me. Right. If you're doing a dihybrid cross, we'd have 4 different phenotype combinations. You'd be doing 4 of these calculations, 1 for each one. Now you just plug the numbers there in, right? So for dominant, our observed, this value here, which is 5474. Our expected is this for the dominant, minus 5493 squared, divided by the expected, which is again this number, plus the observed for the recessive phenotype, 1850, minus the expected, divided by the expected. I'm going to let you work that out while I take a picture of this, because I'm, as you can see, running out of space. All right, you all done? Cool. Give me some numbers. We're going to kind of work upwards. What's the first number for this? First value for this calculation? Going to be what? Ooh, like, what's that going to be? 21 squared divided by 5,493, I think. Or was it 19? Nah, can't work that in my head. $19. Oh, well, that was a good guess. Right, 19 squared divided by this, please. Thank you. 0.657. This one, please. Also 19. Cool. 0.197. Okay, add those two together and you get what? I'm sure you can do it quicker than I can. Oh, where do you get 0.26 from, Adriana? Not sure. Go back and do these. Okay, give it a go. So I'm just going to put this up while, oops. Pay attention. There you go. So for how many of these you do it, you get these values, you add these together. That's what the sum of bit means over there. That gets you your chi-squared value.
Professor, I think for the first value, yeah, it's supposed to be 0. So 0.0657 instead of 0.657.
That's okay.
And so I think the answer is supposed to be, because I got 2.63.
Okay, that's cool. So I'm sorry, Adriana, you are actually completely right. Thank you, everybody, for the check-in. Awesome, great. That's how you figure it out, right? So it is what, 0.26? That's all right, Lindsay. Don't worry about it. That's why we do it in class. Is it 0.26? Awesome. Thanks. Yeah, that's why you practice. Practice, practice, practice. Never hurts to diary and just kind of run through it again until you're really confident that, oh, thank you, Maya, that what you got is right, makes sense. Okay, so now what's the next step? Next step here, I know it's not in order, but you know, we'll figure it out. Calculate the degrees of freedom. Freedom, right? DF. And that is n minus 1. Number of conditions, phenotypes, whatever it is that you're actually measuring. So here we have 2. So the degrees of freedom will be 2 minus 1. And so in this case, and it will vary, our degrees of freedom is 1. Now, essentially, this is used to take into account that the more of these calculations you do, the more conditions you have, the larger your chi-squared will be, simply because you're adding more things together, right? So this is used to take into account of that, right? That's kind of where the degrees of freedom come in. It's n minus 1 because it's an incomplete estimate of the population. And then 5, calculate the p-value, right? And this is the probability that the result you got occurred by chance. That's what the p-value means, represents. So a large value is a high probability that the result you got occurred by chance, i.e. random noise. Very small p-value means it's very unlikely that the result you observed was by chance. Oh, sorry, Adriana. Degrees of freedom is n-1. So if you look at 20 different phenotypes, for example, 20 minus 1, it will be 19. Here we're just looking at 2. So it's 2 minus 1 equals 1. So I'll write that out. Make it a little clearer. Awesome, thanks. Okay, so you can't calculate the p-value. Well, I guess you can. I wouldn't want to. Yourself, what you do is you use a table or a chart, right? Actually, let's write this out. Use a table. Find the DF, right, for your particular experiment. And then you see if the p-value is less than 0.05, right? Because that is the, I don't know, line of significance. Under that value, so as I say 0.01, it's significant. Above that value, it's not significant. So let me show you that table. Doopy-doopy-doo. Here we go. Okay, so I like using the table. Table is a little bit less accurate than the chart. You can use whichever you wish, right? You just have to use one. And so what you do, well, this is how I like to think of it. You look at the top, right? So this is our p-values. And the one that we care about is this 0.05, right? The one right under the ability of probability. That's what we care about. Oh, there's something in chat. That's correct, Maya. Above 0.05, this number, it's not significant. Below that number, even if only a little bit, it is significant. Yeah. Essentially, what this line is saying, or this value, is that this is the probability of getting that result by chance. The probability is 1 in 20. Right, and so that's just someone decided, you know, that's where we're going to draw the line. So if the probability is less than that, say 1 in 100, let's say, it's significant. If it's higher than that, say 1 in 10, it's not. So you want to find, well, this is what I do anyway. You find the column, right? So this is the one we care about, that's 0.05. And then you find your row, right? Your row is the degrees of freedom. So we're going to be working with the top row because we only have a degree of freedom of 1, right? Because we have 2 different conditions. And now you look over here and see, is my chi-squared value, which is this, is it larger or smaller than the value that my cursor is pointing at? 3.84. Is it larger or smaller? Yeah.
Smaller.
Anyway. It's what?
Smaller.
It's smaller. Right, exactly. It's a lot smaller, in fact. Right, and so it's smaller than the smallest number that we actually have on the chart, which is great. So that means that it is, the p-value is greater than 0.5 for this chi-squared value. Right. And so because there's essentially a 1 in 50 chance, 1 in 2 chance, sorry, not 1 in 50, 1 in 2 chance of getting this result randomly, it's like a toss-up, 50-50, like a flip of a coin. It's not significant. And so what that tells us is what we actually observed, these numbers down here, they do differ from what we expect, which are these numbers, but they differ just from random noise. There's no actual reason why they differ from what you expect. If you were to repeat the experiment again, you may have observed numbers which are the other way around. You know, oh, sorry, let me go back. Right, so these are our observed numbers for that example that we did. These are expected numbers. Give me a second, Rachel. I'll kind of go through that in a little bit. Just let me finish this bit. And so if we repeat this experiment again, next time maybe our observed for the dominant would be a little bit higher than what we expect, and our observed for the recessive would be a little bit lower than what we expect, right? Essentially, it's just random fluctuation and noise, right? And so just want to write out something before we get to that question, Rachel. Odalise, did that answer your question? The numbers that we're talking about? Cool. So to properly report a chi-squared test, you have to have the chi-squared value, the degrees of freedom, and the p-value. You've got to have those three things. P-value alone, maybe, I guess, but really you should be putting the three things down. So chi-squared is 0.263. Degrees of freedom equals 1. P-value equals 0.5. And that is a chi-squared test. Just let people write that down. I love having this big board. It wasn't intentional that I didn't get through this on Thursday, but it was a happy accident, I guess. Okay. So to get to your question, Rachel, and it's a good one. And it's also one which kind of really gets at what this test is about, right? And so really your hypothesis is that if you do a monohybrid cross in, let's say, between round and wrinkled peas, right? Or the P-shaped trait. Your hypothesis is that, those alleles segregate randomly, and as a result, you should get a 3-to-1 ratio in the F2, right? That's the hypothesis. And so if your hypothesis is correct, you would expect, you wouldn't expect exactly a 3-to-1 ratio, because biology is never that neat. That's why we have a chi-squared test. But you would expect that the difference between what you observe and what you expect would be quite small, right? So, and actually, let me type this out because that will help to have it as a kind of record. Basically, because you'd never expect to get exactly what you expect. So that's a little bit of a clunky way of putting it. Right. So when you sum those differences for each thing that you observe, you should get a small chi-square value. Does that make sense? Because there will be a difference, but that difference when you add it all up will be a small one. If your chi-square value is small, and really what we mean is smaller than the value in the p-value, oops, period is necessary there. So really what we mean is that, you know, for any given number of degrees of freedom, if your chi-square value is smaller than the one in the p equals 0.05 column, essentially that means that whatever difference you did observe is down to random chance. That's correct. That is exactly correct. So that's why it's so important to understand what exactly you're testing with a chi-squared test, because the various numbers, right, don't have inherent like properties, essentially, like big numbers and small numbers, whatever. So really it's the number, chi-squared number represents the difference between observed and expected. And the p-value is calculated using that and the degrees of freedom. So, and the bigger the p number, the higher the probability that that difference that you observed occurred by chance. So just to illustrate this. Yeah, exactly. That is exactly correct. Let's do a quick example, because I do need to get on to mitosis, because I have to get through this and meiosis before our exam next week. Right, so a quick example. Did a monohybrid cross. And you observed 127 grumpy individuals and 97 happy individuals. I don't know why I always put grumpy as being the dominant trait. I mean, maybe that reflects on me. What's the chi-squared value? The degrees of freedom and the p-value. All right, have a crack at it and ask me questions if you get stuck. Going back, and seriously, you know, I kind of make fun of my multi-point plans for success, but if you follow those points, you'll always get it right. Right. So go back, look through your notes, Sorry, I can't kind of post up the pictures easily. And follow those through, right? One by one. Don't take any shortcuts. I'm just going to take a picture while you'll do that. Ooh, that's a bit. Oh, clean up. And I'll put up the thingamajig table. All right, should be finishing up pretty soon. Just making myself a note to double check the chi-squared test questions in the exam. Make sure that they'll work out of order. Okay, so need to kind of move on and work through this. You should have finished it already, give or take. So hypothesis stays the same, right? So monohybrid cross. What would the probabilities for the dominant, let's say, grumpy and happy phenotypes be? What are the probabilities, right? So this comes from your hypothesis. Right, 3/4 and 1/4 grumpy and happy, right? We're assuming that it's that way around. What is the total number of observed individuals? What's your total observed? 224, great. And if just, kind of double check each other's work in a sense, right? So what would you expect? How many grumpy individuals would you expect if grumpy is the dominant phenotype? Right. 168, exactly. Good job. And the happy individuals? 56. So you can actually take a shortcut, at least with 2 traits, you can just subtract the dominant from the total. That's a bit of a shortcut, so don't try and do that for more than 2 different traits, right? Otherwise you'll get in trouble. Okay, so those are your expected values. Now calculate your chi-squared values. So what's the chi-squared value for the dominant phenotype? It should be what? 127 minus 168 squared divided by 168, right? 10.1. Awesome. What's the value for the recessive phenotype, the happy phenotype? 30. Shit, that's a big number. Okay, add them together. That's your chi-squared value. So chi-square equals 40.01. Okay, so you're nodding heads. Must mean that was all good. Degrees of freedom. That's easy. 1. Good. Okay, so now we take that value, right? So again, we're looking at this column here, the 0.05 column, and the row we're looking at is the top one. So is our value larger or smaller than 3.841? It's larger. It's considerably larger, right? In fact, it's larger than the largest value that we have on the table. There's a bit of an extreme example, right? So your chi-square test is chi-squared of 40.01, degrees of freedom 1, p-value is less than 0.001. And that is essentially a 1 in 1,000 chance of this result occurring randomly. Right, so it's really, really unlikely. It's actually probably less than 0.0001, like 1 in 10,000 would be my guess. It's really, really unlikely that this result occurred by chance. And so what that tells you is that your expected values were wrong. It is highly significant, Maya. Because you observed is what you observed, right? It's neither right nor wrong. It's simply what you observed for that experiment. So if there's a huge difference between what you expected and what you actually observed, that tells you that what you expected was wrong. And given that your expected values come from your hypothesis, that means there's something wrong with your hypothesis. So in this case, I'm going to go and look back at those numbers. Here's a good question for everybody. What do those numbers more closely fit in terms of the crosses that we have done? Geez, we're actually going to run over time, unfortunately, on this. What kind of cross do those data look more likely to have come from? So far, we've learned about 3. Monohybrid, dihybrid, begins with T. It's what you do to try and find out the genotype of a dominant individual. Yeah, exactly, Lindsay. It's a test cross. Well, maybe, right? So then you would repeat your chi-squared with a hypothesis that you're actually doing a test cross and that you have a heterozygote as one of the parents and a homozygous recessive the other. Repeat the chi-squared test and see what you get. So Rachel, that all comes down the last part of that. Why is, if it's less than 0.05, it's significant? In some ways, that is a arbitrary value, right? There's nothing inherent to 0.05 that would make it more interesting or not than a slightly bigger or slightly smaller value. But what it means is that for p-value, of 0.05 equals a 1 in 20 chance that your result occurred randomly. So a fairly small probability. And if it's a small probability occurred by chance, therefore something else, in this case, wrong assumptions or wrong predictions, is going on, right? It's not just random biological noise. It's actual biology, yeah. So that's the thing with the p-value. The smaller it is, the happier we are, right? So you have to think about what is it that you're actually testing, because there'll be other kind of statistical tests that you do where the outcome may be smaller or larger, right? But it's the p-value that really matters. because that's a measure of probability of getting that result by chance. So if you did it again and it occurred by chance, you'd expect a different result that also occurred by chance. If something was actually going on, then it's unlikely you'd most likely get a similar difference again. Because what is going on is not due to chance, it's due to some aspect of biology, whatever that might be. That help?
I just had a quick question.
Uh-huh.
So if we were to do this chi-square test as like a test cross,
Mhm.
would we, like, if it was a question on test, would we be given like the alleles if we know if they were like homozygous dominant or heterozygous or?
Yeah, I'll give you
Okay.
all the information that you need to know. Either I'll give you the genotypes or I'll tell you it's a dihybrid cross. Or I'll give you all the different phenotypes and say the phenotype of the heterozygous or the F1s. So from that, you can figure out the dominant phenotypes. Yeah, this is definitely one of the kind of question that will be kind of at the higher end of the scale of kind of testing you or pushing you, right? Because I might not give you all of the information, but I'll give you enough information to be able to work out everything that you need if you understand what a dihybrid cross is and how to do a chi-squared test and things like that. Okay, any other questions? Cool, all right, so I strongly recommend you practice this, pardon me. And there are plenty of examples here. I think there are, if you look back in the Mendelian cross stuff, there might be even 1 or 2 for dihybrid cross, right? Because I may be giving a question on a dihybrid cross, right? Which is more complicated than what we've just done as a example, right? So practice this. It's not too hard to do when you understand how to do it and you've had some practice at doing it. Okay, so don't leave it to the exam to figure out whether you know how to do this or not. That wouldn't be the best time to find that out. Okie dokie. Let's get back up here. Right, so this is going to have to be a little bit of a wild ride. Get out of it. Because we're running a little bit behind. So, mitosis, right? The principles of cell division, right? So I'm going to write down some kind of vocabulary essentially, right? Which is important. So mitosis is often used to refer to cell division, but it is really the division of DNA in cell division that equals 2 identical diploid daughter cells. So there's a little bit of ambiguity, right, in terms of how we talk about this stuff, which you need to be comfortable with and aware, because we just kind of slip into that because it's obviously easier to say than this. You know, because technically cell division is both mitosis and meiosis, right? It's the process by which one cell divides into 2 cells. Mitosis is the cell division that results in 2 identical diploid daughter cells. Meiosis is a whole nother kettle of fish, right? Quite different. And you've got to understand mitosis, because otherwise meiosis will just be a, right? Mitosis is actually very similar in some ways to meiosis 2, like the second round of meiosis. But I'll try and be precise in lecture, but I guarantee I'll be precise in the exam as to what exactly I'm referring to, right? Because that's really, really important. And there's an awful lot of precision in language with cell division. You have to be very careful about what it is you're saying, particularly when it comes to meiosis, because there's a very big difference between meiosis 1 and meiosis 2. Really big differences, right? In comparison, mitosis is a walk in a park. You know, it's easy, right? We have homologous chromosomes. And then some of this language will be language that you're familiar with. That's fine, but it's worth kind of typing out because otherwise you might be lost if you don't. Essentially, those are two copies of two similar copies of the same chromosome. Same genes, size, length, etc., but different alleles, potentially. One from your father, one from your mother. Now, that is different to, quite significantly different, shouldn't actually use that word, notably, maybe, to sister chromatids. Those are two exact, one hopes, unless there are errors in DNA replication that weren't caught, of the same chromosome. That you get after DNA replication. So there's very important difference between the two. Homologous chromosomes, one from one parent, one from the other parent originally, like way back when you were a fertilized zygote. They are the same length, same genes, maybe different alleles. So they're not identical. They're almost, like really close, very similar, but they are not identical. So for example, the homologous chromosomes in my kids, funny to think of it that way, are likely to have quite a few differences because my wife is from South America and I'm from England, right? Very different kind of populations, essentially. Now, sister chromatids are exactly the same, and they're sister chromatids while they're stuck together, right? After DNA replication. As soon as they get pulled apart, they become chromosomes. And that occurs in anaphase, or a part of mitosis or meiosis. Another important word, cohesin. It's a protein that sticks the sister chromatids together until, pardon me, anaphase. And separase protein that digests cohesin to allow anaphase. And cohesin and separase play really important roles in the timing of anaphase. And also in kind of, in a sense, the alignment of chromosomes on the metaphase plate. A couple of last ones. Kinetochore is a protein structure there that assembles on the centromere. And that's where spindle fibers attach. And spindle fibers are basically a protein polymer called microtubulin. That helps pull the chromatids apart. Let's just double check exam time. We might have to. Yeah, we'll have to see. So we may have to punt exam 1 next week back to Thursday, so the 16th. We'll see how we get on this Thursday. If we get through everything that we need to in meiosis, which is possible. But if we don't, then I'll need to move the exam back because I can't test you on stuff that we haven't covered. It's kind of rude. All right, so those are key words, right? And they are very important ones because they are really the ones that get at the important stuff, right? The mechanism of mitosis and meiosis, and also less so during mitosis, but definitely in meiosis, what goes where and when, right? Which is a really big part of how meiosis works. Okay, beak. So this is essentially a view of condensed chromosomes, right? So it's only during prophase, the actual first part of the division of DNA, that your chromosomes look like this. Where's the Y chromosome gone? Oh, that's kind of unfortunate. Oh no, that's a female, 2 X chromosomes. So we've got 23 pairs, so we've got 46 total. 23 pairs, so we've got 23 from one parent, 23 from another parent. And they are the same size. Rack them up, they look about the same. They have the same genes. They do not necessarily have the same alleles. It just depends on your genotype for that gene. So this individual is heterozygous, has 2 different alleles. It may be homozygous, dominant, or recessive for other genes. You know, depends on what you're looking at. So this is something we don't have time to get into today, but we will cover on Thursday. This is a really important part of cell division, right? The cell cycle, and particularly the checkpoints that control it. So there are different phases, right? So we have interphase here, which is really about all the preparation that needs to be done to be able to divide your DNA and your cytoplasm. And then the M phase, which is really the part where DNA is divided, and then cytokinesis, where the cytoplasm is divided. These checkpoints are incredibly important to make sure that your cells, A, divide when they should, right? During development mostly, or maybe stem cells in your skin or gut or whatever, right? And also, when these go wrong, that essentially leads to cancer. Right, so the control of these checkpoints is incredibly important. They control when and where cells divide, but they also act as a checkpoint, literally, to prevent damaged cells or cells without enough energy or cells with damaged DNA from dividing, like as a protective mechanism for the organism as a whole. Right. And quite often, cancer is the result of either, well, it's a result of a lot of different things, but typically things that result in broken checkpoints or broken DNA repair mechanisms or overactive growth signaling. Those are the main kind of underlying causes of cancer. Okay, so that's that. I've got to throw some food down my neck before lab. We'll continue this on Tuesday. I'm going to be a little bit abrupt because I've only got like 10 minutes to get stuff done. Read through the rest of this. We'll be covering it really quickly on Thursday because I really want to try and get through all this so our exam schedule doesn't change. And then we'll go through the rest of it on Thursday. All right, see you all soon.